how many of you are here who is finding difficulty in revising Unit 4, that is Cell Communication and Cell Signaling under CSIR Net Life Sciences syllabus? So if your answer is going to be yes, then I'm back here with some important tips and tricks that will really help you to revise this Unit 4 in a short period of time and also in a better way. So hey friends, this is Riya Khilmani from Biotechnica. So before proceeding with this video, I would like to request all of my friends to stay tuned and connected till the last part of it, so that you can get a better tips and tricks out there and also be benefited at the last. So getting with no more delay, let's proceed on to the video. So the very first question that it comes is what is this Unit 4 about? So this Unit 4 is all about cell communication and cell signaling. How does a cell communicate? What are the signal transaction pathway that occurs between and within the cell? We also do a study about immunology here. What are the immune system? What are immune cells? And what is the immunology behind these immune cells? So if you see the previous year questions, you will find that this Unit 4, it is asked along with Unit 2, that is Cell Biology of CSIR Net Life Sciences syllabus. So it is interconnected with Unit 2. So I suggest you all to not skip this unit. So now let us look into the syllabus, what all topics are included under this section. So if you see, there are 5 subparts that are included in this unit. First is host-parasite interaction. Second is cellular signaling. Third is cell communication. Fourth is cancer. And fifth is immunology. So yes, so now coming to marks distribution. What is the weightage of this unit? Like if you see the previous year questions, so on the basis of previous year question analysis, it is found that the weightage of this unit for part B is 14 to 15%, and for part B it is near about 14%. So if you see the CSIR net exam pattern, there are 3 core sections in that exam. So first is part A, that is general aptitude. That is a common topic for all. Part B, that has a direct subjective question related to a particular topic. And for part C, it is a detailed and experimental based topic related to that particular subject. So for part B and part C, this is the marks distribution. So yes, so this is the reference book that you can refer for your study as well as for a revision purpose. Now, the most important thing, what would be the study plan? What is the need of this study plan to revise this Unit 4? So here I've just made a one-day schedule for you. If you want to extrapolate it, you can do it according to your speed of revision. So let us see how to manage this revision in just a single day. So here you can dedicate your 2 hours for part A, for part B, for part C, as well as part D. For part E, which is immunology, it has more number of topics comparative to other subparts, that is A, B, C, and D. So here you can give 3 hours for a revision. And once you are done with all your revision, now it's the time to implement all your things that you have learned. So for this, you can practice last 10 years previous year questions so as to get a better understanding of a subject and how the pattern of questions that are asked in exams. So now let us talk about the tips, what is needed to revise this unit. So the tip number 1 here is comparative table. You can make a comparison table for the topics that are listed here. Like if you want to study humoral and cell mediated immunity, so for this you can prepare a comparison table. Like what are the cells that are involved in these kinds of immunity? What are the mediators that are taking care of this immunity? What are exotoxins? What are endotoxins? Which bacteria is responsible for secretion of these kinds of toxins? Which toxin is heat stable? Which is more stable? So you can just do a comparison study of these topics. What is DNA and RNA viruses? Like, what is an example of double-stranded DNA virus? What is an example of single-stranded RNA virus? Because many a time CSIR focus on the examples of these viruses. They just ask a question, which of the following is not an example of this DNA virus? So accordingly, you have to pick a suitable option according to that. You can also do a comparative based study of the primary as well as secondary immune response. What are the name of antibody that gets heightened at the time of primary response? What is antibody that is responsible for causing the secondary immune response? What is a lag phase in primary response? What is a lag phase that is there in secondary immune response? You can also learn about the pathogens that are there that causes diseases in plants as well as animals. So there are different types of pathogens, there are different kind of microorganisms that are causing diseases in humans, in animals as well as plants. You can also do a comparative study about the genes that are responsible for causing the cancer, because there are some genes that promote cancer. There are some genes that suppress the cancer. That is what called tumor suppressor genes. So you can do a comparative study of P53, retinoblastoma, and many other genes that are involved in cancer pathway. You can also do it for normal and cancerous cell, so as to study the properties of these cells. What are the properties of these cells? How they are different? What is anchorage dependent? What is non-anchorage dependent? What is apoptosis? What is necrosis? What are the processes that is making one cell to be apoptotic? What are the processes underlying, like if it is chromatin condensation, which cell is causing blebbing? So all these things you can do on the basis of formulating the comparative tables. You can also learn about the different types of antibody that we have. You can do a comparative study for 5 different kinds of antibody like IgG, A, M, D, and A. You can revise them on the basis of their structure, function, as well as if these antibodies are cleaved by certain factor, then what would be the number of bands on the STS electrophoresis? Like this, you can revise these antibodies. And also you can do it for MSC molecule 1 as well as 2. Now, what is this MSC molecule 1? It is a part of immunology. So you can compare these MSC molecules on the basis of this comparison table. So the tip number 2 here is concept wheel. So what is this concept wheel about? So this is an innovative idea that is designed by Biotechnica itself. So what is this about? In the concept wheel, if you see the name of the topic that is there in the middle, and this topic name is surrounded by the information that is related to that particular topic. So you can revise things on the basis of this concept wheel also. So if you want this concept wheel to be purchased, the link is there in the description box. You can refer it for any of the topic or for any of the unit. So next, what is tip number 3? Tip number 3 is here, creating mind maps for your easy interpretation. Now, what is this mind maps? What is this mind map? Mind maps helps to understand better the interconnection between things. It provides gist of a topic at just one place. So you can do it for signaling pathway. Like suppose if I'm considering the example of GPCR signaling pathway. So you will write the name of that pathway in just the middle. That will again be surrounded by the information related to it. And you can relate and interconnect this GPCR to all other pathways that are activated with the help of GPCR only. Like I'll give you an example for this, like PLC beta pathway, like there are some other pathway like cholera toxin, pertussis toxin, how rhodopsin toxin is activated with the help of GPCR, how it is regulated. So all things you can just interconnect all these things with the help of mind map and revise it. So you can also do it for cancer. Like cancer, you can just write the topic name in the middle and the features of cancer cell and how many genes are there that are causing this cancer, and what are the pathways that are regulated at the time of cancer, say how cell cycle checkpoints are regulated. You can also do it for antigen antibody interactions. Like there are many number of antigen antibody reactions like precipitation, what is agglutination, what causes the clumping of RBCs, what is octilinear radial diffusion, what is immunodiffusion. So you can prepare a mind map for all these topics. So I will say keep it short and precise. tip number 4, figure-based study. So why it is important? Because it is less time consuming. So if you are doing the figure-based study, so these figure-based questions, they are asked every time in CSIR net exam. So they just give you a schematic representation of a process, and there you have to deduce which pathway is this. So for this, figure-based study is very important. You should not skip this. What is the tip number 5? You should also prepare a common charts for preparation. Now, what is this common chart? What are the different topics that you can refer and you can revise with the help of these charts? So the first topic over here is you can revise enzyme secretion and the pathogens related with it. Like there are many different kinds of organisms like staphylococcus, like which kind of enzyme does it secrete? Leukocyte is released by which kind of microorganism? Streptokinase is secreted by which kind of pathogen? Like this, you have to prepare a chart and revise your concept. You can also do it for the different types of TLR. What are different types of TLR? Like I have put this table over here for a reference. What are the different types of TLR? What are the PAMs that is specific to TLR? Like many times these are asked and match the following form. So you should do it on the basis of this chart preparation. Now, what is this immunity? For which kind of antigen, for which kind of autologous antigen the antibody is developed in the body, like whether it is Graves' disease, your myasthenia disease, you can just list out the name of the disease, name of the autoimmune diseases, and along with it the name of the antibody that is formed for the autologous antigen. That's it. This is done for autoimmunity wala part. Now, what is this hypersensitivity? You can do it for hypersensitivity also. When the immune response gets hyperactivated. So there are many diseases that are occurring inside the host body when our immune response is hyperactivated. So you can refer, you can revise all these topics on the basis of these charts. Now, what is immune deficiency diseases? There are many different types of immune deficiency diseases in the body. So you can just learn the example of immune deficiency diseases and what all cells are there that are getting completely lacking in this immune deficiency diseases. So the major example of immune deficiency diseases is SCID, severe combined immunodeficiency disease, where lacks a B and T lymphocytes together. So you can do this study also. So what are the different types of vaccines? What is toxoid vaccine? What is live attenuated vaccine? What are examples of this vaccine? So just prepare a common chart for them and have a glance just at the time before the exam, and you will definitely make it. Tip number 6. Now there are some topics in Unit 4 such as extracellular molecules that are asked in match of the following form. Like which all topics are there? Extracellular molecules include selectin, cadarin, immunoglobulin superfolds. So all these topics, they are just asked in match the following form, and you have to match according to their functions. Structures and functions are only asked. So how are they asked? They are very direct question. They are easy to and they are scoring. So you should not skip that too. Now, what is tip number 7? You should always do a graph related study. Now, why this graph related study is important? So whenever you are studying immunology, you will always find graph there. So in CSIR exam, it is always asked, suppose when any bacteria or virus enters into the body, then what will be the immune response that will generate it at the time of this infection? So what will be the latency period? What will be the lag period? So for this, graph relative study is very important in order to find the answer to this question. What is tip number 8? Revise some topics that are merged and asked along with Part C questions of cell biology. As I already mentioned earlier, this topic, Unit 4 questions, this is asked along with Unit 2, that is cell biology. Like what are common topics that are asked with Unit 2? There are some topics like cancer as well as cell cycle. What are the different checkpoints? What are the regulation of cancer at different stages? What are the defects in the signaling pathway that are leading to cancer? So you can do study about all these topics, because these topics are merged and asked with Unit 2, that is Cell Biology of CSIR Net Life Sciences syllabus. Now, what is tip number 9? Always try to make use of a flashcard. Now, what is this flashcard? It provides a way to overcome difficulty. You can just make a sticky note for it, for important points. Just jot down your tough points there and paste them in front of your study walls. So it is very helpful in remembering difficult topics that you forget every time. So yes, so now what is the final note for today's lecture? So I would like to suggest all of my friends to attempt only those sections that you have already recalled and revised. Do not attempt section that you have not went through. Never attempt these section, and always solve question if you are sure about it, because every part B question there is a +2 for a positive answer and there is a -0.5 if you are getting it wrong, and for part C it is +4 for every right answer and it is -1 for every wrong answer. And so you should make a final note that you should only attempt those sections that you are really confident about. So I would just like to conclude my topic by saying this: don't be concerned about reaching the destination. Never be concerned about reaching the destination. Just enjoy the journey and the destination will take care of itself. So thank you once again for watching this session. Hope you liked this video. If you like this video, do not forget to like, share and subscribe. Also share to your friends and colleagues whosoever is attempting this CSIR NET exam. So you can also find a free mock test link that is there in the description box. So you can open that link, you can download the mock test paper and you can just attempt it and rate your success. So thank you once again for watching this session. Till then, bye-bye, take care and have a nice day.